have a handout in case you want to do this incognito, directions on how to do it. If you need for me to show you how to do that, that would be a good time to come see me in an office hour, but I'm not gonna spend any time on that now. All right, last time we talked about some basic chemistry, about how some atoms will combine to be 288. The 288 is the electron. Zoom out. Okay, this is important to know. Because of that, oxygen wants to form two covalent bonds and two hydrogens with protons on one side. The rest of it is all the electron shells, which is negative. So we have a north pole different from a south pole, that's polar, and things that are like water, love water. So things that are polar are hydrophilic. Here we have methane, it's completely symmetrical, there are no poles, it's nonpolar. Things that are like it are hydrophobic, including if instead of just one carbon, it's a whole long hydrocarbon chain. Does that make sense?
Yes.
We talked about that with a phospholipid. Are we ready to move on? Okay. What were the 5 things, the top 5 things included in cell membranes, the fluid mosaic model? What's first? Phospholipid bilayer. What's second? Protein bolus. What's third? Cholesterol. What's fourth? Cytoskeleton filaments on the inside. What's fifth? Carbohydrate groups on the outside. Okay? Then we had a slide that looks something like this, where we talk about selective permeability. Remember this animation? Doesn't say so on the slide, but you should fill in the blanks and write selective permeability. That little phallic looking thing represents things that are small and uncharged. The hexagon are things that are larger or have a charge. Charged means it's an ion. You guys remember when I went over what ions are? So if it's charged, it's an ion or electrolyte. And then these things are huge, like a large protein. So small and uncharged things are things like water, CO2, O2. Larger things are things like glucose. Charged things are things are ions, electrolytes, which are synonyms for us. And the huge proteins don't ever go through, they're too big. So if you look at this animation, and the first bullet, things that are small and uncharged, like this little arrow, can go through the lipid bilayer. The hexagons, the glucose, has to go through a protein channel. Here's the small uncharged thing going through the bilayer, but it's got to be the right protein channel, not that bottom one, but that one. So I'll say specific. That means never go through. All right, everybody understand that? So first bullet is the little arrow of this guy, goes to the lipid bilayer. Things that have a charge or a little bit larger like glucose have to go through the specific protein channel. Bloop, right there, that's the second bullet. And then this guy here never goes through. As we're gonna talk now about transport processes, it's really helpful to keep this organized in your head. My anatomy students, does this look familiar at all?
Yeah.
Same freaking slides. All right, we have to talk about some terms. We have a liquid mixture. The whole liquid mixture is called a solution. It has to have a liquid medium we call let's see if this works. Solvent. There it is. In biological systems, that is always water. And then the stuff in the water, that is solute. Okay, everybody got that? Now, if this, all the solute was up on this end of the beaker, this would be the high concentration of solute, that's why it's red. Down here would be the low concentration of solute. And to move from high to low takes no energy. To move from low to high does take energy. And that's what the third bullet is talking about. If you're going from high to low, that is down the concentration gradient. So just think of it like bequest the grade, concentration gradient. It's the same root word. Everybody got it? You could be up in Santa Margarita, turn your engine off and coast to San Luis. You'd be a hazard, but you can do that. Passive. No gas is being burned. But to go from San Luis to Santa Margarita, you gotta burn gas. Okay? And that's cellular energy, and that cellular energy is always ATP. I'm gonna talk about this more in a bit. But that's what you need to go uphill. By the way, lecture exam 1 is made up, the most points are on 2 topics. One is ATP, and the other is DNA. I'll talk about that more later. So when we talk about ATP, you should perk up because that's a big point thing on lecture exam 1. Okay, everybody got this? Third bullet, what it means? So if you're gonna go from high to low, passive. If you're gonna go from low to high, you gotta burn some ATP. All right, so let's talk about passive transport processes first. Basically, what I'm doing on the whiteboard is what I would do as a student with my Nerd 4-color pen. Because it allowed me to connect dots. And dots I connected, I would understand. And what you understand, you don't have to memorize. Let me repeat that, because this is going to be huge for you later on. You think this is a lot of content here? You guys are destined for classes that have way more content than this. How are you going to handle it? You're going to shoot for understanding it. Because what you understand, you don't have to memorize. I would pay attention to that. All right, so we're going to talk about passive processes first. So I would always write outlines, I'd use different colors. And so this is, the thing that this has in common, all of these, is no ATP needed. For most of them, it is going to be about going downhill, but not all of them. So this is always constant, no ATP needed, but it's going to go from high concentration to low concentration. Okay? So let's start here with A. Diffusion. One word diffusion. One word diffusion is about the solute moving from high to low. Okay, so on this animation, the purple dots are like the red dots. It's going from high to low. Everybody see that? The animations don't cycle forever. Now what you notice is that if you look at the big animation here on the right, they're moving in all directions. The movement is completely random. But if you start with more of these purple balls on this side, more of them are going to move to the right than the balls on the left are going to move to the left. Okay, so look at the big arrows. Until they become even. When the two arrows become the same size, you achieve what is called equilibrium. So right where it says no net flow, write equilibrium. There's the same number of balls going to the left as going to the right. Equilibrium. But when you start out with more of these balls on the left side, more of them are going to move to the right. See? Until it's even. What's key here is that these purple balls are moving right through the lipid bilayer. Right where it says this. So that's why I've used my NERV 4-color pen and go like this. That's simple diffusion. One word diffusion. Solute going to the lipid bilayer, just like that little phallic arrow. Everybody got it? Okay. This is, your textbook doesn't call it net flow, it calls it net flux. But this figure is showing you how if you have all the balls on this side, more of them are going to go to the right until it's even, and then it reaches equilibrium. Equilibrium in this case is this middle line right here. Everybody understand that? Or would you rather just read the textbook and understand it from the author's words? Everybody got it? Then we have osmosis. Osmosis is also passive, but this is about the water. It's moving from high to low. High water concentration to low water concentration. So here when I'm saying high to low, it's about the water. For example, in this beaker, this would actually be the high water concentration side, and this would be the low water concentration side. Because this is 100% water down here in the bottom right. It's not 100% over here. Everybody got it? All right. This is what you want to write next to the slide. Actually, a couple things. In order to see osmosis, you need two things. You need a selectively permeable membrane. One, you need a selectively permeable membrane. Two, you need a solute that can't cross it. In order to see osmosis, you need a selectively permeable membrane and a solute that can cross it. Okay, so if you look at this animation, in the middle is the selectively permeable membrane, and these little green dots, you'll notice, will bounce off, but you can't cross it. So we're going to see osmosis. The only thing that will cross is going to be these little wing guys. And those little wing guys is this, the water. Okay, so watch the animation here. See how the green balls just bounce off of it? But it's the little wing guys that cross the lipid bilayer. Everybody got it? Does everybody understand what osmosis is? Okay. This is also going through the lipid bilayer because water can cross the lipid bilayer. So this is also through the lipid bilayer. By the way, on that first animation, what are the two things that you need to see selective permeability? What do you see there in the middle of that animation? In the middle of the animation, what do you need to see selective permeability? What's right here?
The bilayer.
The phospholipid bilayer and protein boulders. Everybody got it? Everybody look at me. Out of those 5 things that you named at the beginning of class, you only need 1 and 2 to see selective permeability. Get that straight for a test question. That's all that's on this animation is the first 2, right? Do you see cholesterol? Do you see cytoskeleton filaments? Do you see carbohydrate groups? What are the 2 things you see in this animation? Lipid bilayer, protein boulders. You only need 1 and 2 to see selective permeability. Everybody got it? Okay. Now, the other thing you want to write next to osmosis is a saying that you will hear even on clinic floors, and that is water follows the salt. Water follows the salt. So, and actually, that's just a saying. The reality is, water will follow any solute that is osmotically active. They aren't all osmotically active, but the ones that are, water will be attracted to. For example, here's water follows the sugar. The little green circles is glucose, sugar. And look how the water is going from left to right. Because it's following the sugar. Everybody see that? Now notice how the water is actually lifting up over here on the right side. What does it take to lift up a column of water in a pipe? Pressure. To actually lift up that column of water takes pressure, and the term is osmotic pressure. Osmotic pressure. Okay. Now, if you look at this animation at the very bottom, you will see that the water is moving from high water concentration to low water concentration. Can you see that? Can I give you a little hint for the lecture exam 1 or the NCLEX? Pick one way of understanding it. Either water follows the salt, or the water is moving from high water concentration side to low water concentration side. Don't switch back and forth, you're gonna get it wrong. Stick to one to understand it. Even after this class, stick to one to understand it. All right, you do need to know these terms. We will actually see this in the lab, I don't know when. This has to do with osmosis, which is the movement of water. The confusing thing for students is these terms have to do with solute, when osmosis has to do with movement of water. But you do have to know what these terms mean. All right, let's just say this is a red blood cell. Kind of looks like a biconcave disc. Let's say, just for shits and giggles, it's 10% solute and then 90% water. Okay, so the combination of the percentages have to equal 100%. Everybody got it? Okay, what this animation is showing you is 3 solutions. Now these names stand for the solution outside the cell, here in the beaker. And what the hyper, hypo, and iso mean has to do with the relative solute concentration compared to the cell. Make sure you write that down somewhere. Relative solute concentration compared to the cell. Well, if the cell has 10, say this is 20. That's more than this. Hypo means less than the cell. So let's go with 0, distilled water. That means less than the cell, and iso means the same. Then again, everything has to equal 100. So 80% water, 100% water, 90% water. Now you gotta tell me which way the water's gonna move and the effect on the cell. We'll cover this in lab as well. Okay. Since this has more solute out in the beaker, water follows the salt, and it's going to go out to the beaker. And the cell's going to shrink when the water leaves it. We call that crenation. So the cell's going to crenate. Here, if there's no salt in here, all the salt is inside the cell, so water's going to follow the solute into the cell, and it's going to pop. When blood cells pop, that's called hemolysis. True or false? On this one here, no water moves.
False.
That's a trick question. Yes, the water's moving, but in equals out. So the cell remains a biconcave disc. Look at the animation. That's too low. Because so much is leaked out, those leaky pipes made the pressure in the pipes low from the leakage. Is this easy to understand? Okay, everybody look at me. This is a disadvantage of me teaching this class. I can take extremely complex things and make it sound simple. Which actually, isn't that what our professor's supposed to do? Aren't they supposed to take complex things and make them sound simple? The problem is that the overriding academic mentality of this valley is that if it sounds simple, it must be simple, so I can veg. It's not simple, so don't veg. Your teacher's just making it sound simple. Everybody got it? Okay. And we got facilitated diffusion. This is also about solute, going from high to low. But in this case, it's not going through the lipid bilayer. It's going through a specific protein channel. So in that animation, these little guys that are going through the lipid bilayer is either diffusion or osmosis, but that hexagon, that's facilitated diffusion. Diffusion, one word right here, bloop. Facilitated diffusion, right here, bloop. Doesn't ever go through. Everybody got that? How this all ties together? Connecting dots? Now, facilitated diffusion, these protein boulders, the specific ones that do facilitated diffusion, show specificity, competition, and saturation. Okay? Everybody got that? Do you want me to explain it in simple terms, or do you want to read it in the textbook?
You could simplify it.
want me to explain it?
Yeah.
Okay. A cell will only have so many of these protein channels. It's not an infinite number, it's a finite number. Everybody got it? So this animation you're seeing, it only is gonna have so many of those. The specificity, and I want you to hear this because I'm gonna say it over and over and over again all semester. To teach physiology at this introductory level, I have to simplify things to the point that what I'm actually saying is not true. I'm gonna spend all semester lying to you, just so that you can get it. And then once you get it, you can actually learn about the complexities that is actually true in our body. Okay, for example, we talked about specificity. First diamond. Specificity is not black and white, it is in shades of gray. In fact, get used to that, people. There is nothing in our bodies that are black and white. Everything is shades of gray. Are we clear? For example, the channel here, that would be, say for this guy, will not let any of these guys pass by, but since this is very similar, potassium, sodium very similar, some of those same channels will actually also allow this one to go through, both of them. Shades of gray. Everybody understand about reality here now? So, can I erase this? This was from last week, right? In fact, I did it with more detail last week. All right. There are ways we can actually measure this transport, facilitated diffusion, or rate of facilitated diffusion. I'm just gonna abbreviate it, facilitated diffusion, okay? It's not fire department, facilitated diffusion. And this is the concentration of solute. Oh, by the way, whenever you see a bracket, that means concentration, okay? So, if you have, say, sodium going through, the more sodium concentration you have on one side, the faster the rate of facilitated diffusion. That's intuitive, that just makes sense. The more concentration of sodium you have on one side, you would keep increasing it, the rate of facilitated diffusion is just going to go up. But, let's go to the third bullet first. You only have a finite number of these channels, and once they're all working as fast as they can, they're going to max out. And when they all max out, the line goes straight. That's the point of saturation. But again, we're talking about sodium here. Because specificity is in shades of gray, if we add potassium, these channels, these finite number of channels, are going to be competing. Between sodium and potassium. And it'll max out earlier. And the reason why they can compete is because specificities and shades of gray. Does that help with the 3 bullets or the 3 diamonds? Do I need to repeat any of that? Everybody got it? Okay. This is showing you again the protein will actually bind to the solute, and that causes it to change shape. And it puts it on the other side, but it's going from high to low, just through a protein channel. All right, let's go over active processes. And this is also usually going uphill from low to high, going up the center line. Now, in anatomy, we only covered primary active transport. In physiology, you're gonna have to learn secondary active transport. Are we ready? Go ahead and fill in blanks there. Again, if you came in late, this lecture PowerPoint is the last one I'm providing you. You're gonna have to print up your own, and make sure you come in with it. You're gonna be scratching and clawing if you come in the lecture without one. If you don't know how to upload them from Canvas and print them out, there's a pink sheet up here in front. And if you still don't know, then come talk to me in an office.
Professor Muhan?
Yep.
Okay, bear with me on my question here. So for a rate of facilitated diffusion, just so I can understand, so if I have a protein channel that has a higher affinity for sodium, right? Sodium is gonna go through, of course, more, but then since it also has a slight affinity for potassium, potassium is gonna facilitate at a, I guess, lower rate?
No.
And still diffuse, or,
Okay, so where the line goes horizontal is the point of saturation.
right?
What this is saying is that if that protein channel has an affinity for both, even though it may have preferential affinity for sodium, which is actually true, if you add potassium, some potassiums are gonna occupy some of those protein channels. So now look at where the line goes horizontal. It's gonna saturate at a lower level because both of them are competing for those protein channels. And they can only work so fast. And you have only so many of them.
Right.
Okay, does that make sense?
Makes sense.
Okay. Any other questions?
Are you saying that the point of saturation is when they're all being used?
And
Or
they're
maxed
going
out?
as fast as they can.
Okay.
All being used, going as fast as they can, it's saturated. Okay? Anything else? Okay, did everyone get this? The key here with active transport is you're gonna utilize ATP. Was ATP covered at all in anatomy?
Yes.
Did anybody not know anything about ATP at all? Raise your hand because seriously, you're gonna be bleeding on the tracks. If you need me to go over this, I'll go over it.
Can you like refresh it?
What does T stand for? Tri, that means three. I'm gonna go over the biologically important macromolecules either Wednesday or next week. But this is an RNA nucleotide. It has a nitrogen-containing ring called adenine, five-sided sugar called ribose because it's RNA. And to be a nucleotide, you gotta have three things. The nitrogen-containing base, a sugar, and at least one phosphate group. In the case of ATP, there are three of them, so it still qualifies. At least means it can have more. So far, so good? This is ATP. Energy is stored in the bonds between the phosphate groups. So if you break off the last phosphate group on this, now you have a free floating phosphate we call inorganic phosphate. This is now ADP plus inorganic phosphate. And this whole thing is called the ADP-ATP cycle. But when you break off this last phosphate group here, you now have energy released for work. The reason why you constantly gotta eat food is your nutrient energy. When you break it down, it's so that your mitochondria can put the phosphate group back on. This is one of the two main topics for lecture exam 1, is this. Was this covered in your anatomy?
Yes.
Man, my students, I'm looking at you, you better say yes, because I know damn well I did this. If this wasn't covered in your anatomy class, you may need to correct your rate my professor, because I know that was suggested of you. Okay, any questions on this? The protein channels, and this also includes protein channels, but since these are transporting it uphill from low to high, these protein channels are called pumps. They're pumping uphill. Okay, now watch this animation. There's that calcium where there's less. Here's the ATP going to ADP and P. Then it's going to drop off the calcium where there's more. It's going from low to high. Did you see that in the animation? Well, you get to see it again. Does that help that I covered this right here? A, D, P, and P. Calcium's going from low concentration to high concentration. There's the sequence. Sequential thinking helps. First, the calcium binds to the carrier protein. That causes that last phosphate group to come off of ATP. It's called hydrolysis. One of those phosphate groups sticks to the carrier protein. That causes this thing to shift in a hinge-like manner, and it drops off the calcium on the side that has more. See the sequence here. 1, 2, 3, 4. Now look at this single sheet handout you got. The essays for lecture exam 1. Turn to the back. I've now covered all the essays in 1.5 lectures on the back. Last week we covered negative feedback loops and positive feedback loops. People, this is copied and pasted from the slides. That's how straightforward this is. Then I said you need to know the different ions and the ICF and ECF. That's number 9. Does that ring a bell from last week? And then I just covered number 10. Read it. It's copied and pasted from the slide. Is that straightforward enough for you? Nothing tricky. Okay. But this is primary active transport. And maybe what you should write next to this slide with primary active transport is the ATP is burned at the site of transport. This blue protein folder, that's where the ATP is burned. That's where the calcium is being transported. Primary active transport. ATP is being burned right here. Boom. At the site of transport. Boom. Primary active transport. Everybody got what I just said?
Mhm.
Any questions? Is this helpful at all that I'm explaining this? Or shall I just have you break up in groups and learn it yourself? You guys know what I'm talking about too. All right, a common, active transport pump is sodium potassium pumps. They're extremely efficient. Because if you watch the animation, it's actually moving 5 ions with 1 ATP. Off to the side of this, you should write 3 sodiums, that's the Na plus, 3 sodiums out, 2 K pluses, that's potassium ion, in. 3 sodiums out, 2 potassiums in. Watch the animation. With 1 ATP, 3 of those yellow sodiums out, 2 of these green potassiums in. With 1 ATP, highly efficient. 3 out, 2 in. Does 3 equal 2? And that is why your cells are negative on the inside. We'll talk about this later when we talk about muscles and neurons. But look up at this picture that I will use the same figure when I talk about the nervous system. Inside the neuron, it's negative. Outside the neuron, it's positive. And part of that, not the whole thing, we'll get to it, but part of it is because of this 3 doesn't equal 2. Does this make sense?
Mhm.
Any questions?
This might be a dumb question, but this seems like to me that it's still at the site of transport though.
It is. This is primary active transport.
Oh, okay, I thought you were.
I haven't gotten the secondary. I just.
Oh, you wrote it.
I wrote it.
Okay, never
I'm
mind.
still finishing primary.
Sorry.
This is still, be sure you see that. By the way, I teach this class the way I would want it taught if I was a student in this class. I don't want to have a question mark of what I have to know. I don't want to have a question mark at what depth do I need to know it. I want it clear. And I want to present it in a way that is in order. That's why if you look at the tops of the slides, you can see the signs of an outline. And right here, sodium potassium pumps is still under primary active transport. If it's organized when I present it, it has a better chance of being organized in your head when you're studying it. And if it's organized in your head when you're studying it, you have a better chance of that stuff not swimming around in your head in front of the Scantron. If I teach it like this, well, then you're out. Okay, 5 ions again per ATP. 3 sodiums out, 2 potassiums in. Now let's go to secondary active transport, which is also called coupled transport. Next to this slide, this is what I would write. The ATP is burned someplace else. That's the difference. And it is a little bit more complex. Instead of just one process, it's actually a sequence of 3. Right. I got to erase from whiteboard. Where should I erase?
The beaker.
Can I erase over there on the right? You guys need to take a picture of the whiteboard on the right? If your phone is up here because you're recording and you want to take a picture of the whiteboard on the right, I'll give you a couple. Taught this class a long time. This topic of secondary active transport just messes with students.
Oh, great.
But I found a way of teaching it where it's actually very simple. Just remember it's a sequence of 3. 1 process first, then 2 simultaneously. Are we ready? One process first, then two simultaneously. Maybe I could just erase this and fit it in here. Okay. And this is also going to help you with this concept of high to low concentration. When we have high to low concentration, that's a concentration gradient. And like the Cuesta grade, it looks something like this. Going uphill, the Santa Margarita, San Luis is down here. Secondary active transport, the first thing that happens is primary active transport. So let's draw a picture of an 18-wheeler. Okay, don't criticize my artwork. And in this 18-wheeler is sodium. The first process is primary active transport,
Okay.
most commonly of sodium. And its primary active transport, right, is going from low to high. Boom, goes up to the summit, Cuesta summit. Have you ever been hiking up there, up the Cuesta summit, Cuesta Ridge hike? That's pretty cool. Anyway, all right, so that happens first. Done. It's now sitting on top of the Cuesta summit. The concentration gradient is there of sodium. Now, down here at the bottom, we have a small pickup truck. And in that small pickup truck is glucose in the bed of that pickup truck. What secondary active transport is we've taken this long chain, hooked it to a pulley to this pickup truck with glucose. Then we have simultaneously, second, this is first, sodium going down in facilitated diffusion and that's going downhill from high to low, but because it's linked with this big chain, the glucose is going up from low to high, and that is secondary active transport. Because this pickup truck's engine is off. It is not burning any gas, it's not burning any ATP. The ATP was burned somewhere else with the semi. Then once the semi's up here, it goes downhill and pulls the glucose uphill. The glucose is still going from low to high, but its engine is off. Because the ATP was burned somewhere else. Sequence of 3 events. First this, and these two simultaneously. So again, we start out with primary active transport pumps, pumping sodium, pumps it uphill. Then another carrier someplace else has 2 binding sites, what are called recognition sites. One for the sodium and one for the glucose. Oh look, I even use the same colors. The sodium is going downhill, and it's powering the glucose going uphill. All the words, but they're not talking about semis and pickups, and chains and pulleys. So let's go over this diamonds. First, you have to have primary active transport. That's on the top. That's the first diamond. Second diamond, now simultaneously you have another protein that has a recognition site for both sodium and glucose. Sodium goes down or along the concentration gradient. Glucose goes up or against the concentration gradient. But it is the sodium concentration gradient that powers the movement of glucose uphill. You read this in the textbook, they're not gonna talk about semis and pickups. So, and you don't have an option to know this or not. You gotta know it. A good teacher will not give you the option of knowing it. But actually will find a way so that you will know it. Any questions? Now, glucose and amino acids are commonly transported this way by secondary active transport. They can either go in the same direction, and I think that's what the last animation on the previous slide showed you, is same direction transport, that's symport or co-transport. So you see the the red and green balls are both going down.
Mhm.
That would be co-transport or symport. Same direction. Same direction, boom. Or they can go in the opposite direction, and that's counter-transport or antiport. So they don't have to be going in the same direction. Any questions on secondary active transport? I kid you not, this seriously used to take me a half hour to explain to students. And then I started using semis and pickups, and, oh yeah, that makes sense. First, you gotta get the semi uphill first, and then because of the pulley and the chain, the other two processes are happening at the same time. One's going downhill, one's going uphill. But the ATP was burned somewhere else. Okay? This is from your textbook, co-transport, counter-transport. Either the same direction or opposite direction. All right, then we have bulk transport. Now in these animations, you will not see ATP being burned. You're just gonna have to trust me. It's actually being burned by sliding filaments. In anatomy, did you guys learn the sliding filament model
Yes.
for skeletal muscle? Well, skeletal muscles are not the only cells that have actin and myosin. Cells that do bulk transport do as well. Do you guys remember how much ATP is burnt by skeletal muscle? It goes through a lot of ATP with sliding filaments. That's how bulk transport is burning. So just trust me, it's there. Bulk transport can either go in or out. If it goes in, it's endocytosis, and it's a sequence. If you do not see solid solute, we call that pinocytosis, which can be thought of as cell drinking. If you can see solid solute, like a bacteria or a virus, that's phagocytosis. In reality, most endocytosis is triggered when whatever's gonna be taken in binds to antibodies. And antibodies are shaped like capital Ys. You see black Ys in this animation. It's binding to antibodies and it's bringing it in. And really and truly, that's most of it is like that. Now, this phospholipid bilayer is very fluid. The same phospholipid bilayer, if you look at this animation, that used to be the plasma membrane on the outside, becomes the membrane-bound vesicle on the inside. Same phospholipid bilayer, same red line. These other animations, there's a purple line. But again, it's the same phospholipid bilayer, okay? Any questions? If it can come in, it can also go out. This is showing you in the textbook, phagocytosis, panocytosis, and then receptor-mediated endocytosis where it binds to the capital Ys, the antibody. Everybody see that? Okay. True or false? Everything that, all your cells are fed by what you eat.
False.
False. For phagocytes, like white blood cells, if you trip and skin your knee and you get bacteria and other things in there, if it's biologic, that white blood cell will have antibodies on it. It's gonna bind to that bacteria, it's gonna draw it in, and it's gonna break down that bacteria and digest it, and make ATP out of it. That white blood cell is fed not by what you ate, but when you skinned your knee. I just think that's bitching, and you guys like. It's like a Hollywood horror movie. Your cells are being fed, you didn't even eat the food. Okay, well, we'll move on. That was underwhelming. All right, so stub, big bulks can go in, big bulks can go out, that's exocytosis. And this animation is really good here on the left because it shows you that what was a lipid bilayer vesicle will just fuse and become part of the plasma membrane. That blue will become red. And by the way, this is how you get these big guys to either go in and out of the cell by bulk transport. Okay? Your neurons at the axon terminals will have neurotransmitters and vesicles. When the action potential goes to the end, you'll get a spike of influx of calcium ion, and that triggers exocytosis. That's how it spits out the neurotransmitters. Okay, let's see. Let me see if you can see this in this animation. See the little flying saucer things in the animation? Everybody see that? Notice it's coming into the cell without ever crossing the red line. It's not crossing the red line, is it? So things like that can still get into the cell without ever crossing the red line, without ever crossing the plasma membrane. You can still get proteins out. Insulin is a huge protein. How does your beta cell spit it out to your bloodstream? By exocytosis. And that insulin never crosses the plasma membrane. Just spits it out by exocytosis. That's, yeah, because insulin is huge. We're gonna write that down here. We'll see later, it's one of the few examples of quaternary protein in the body. Okay? Any questions on transport processes? But these things like in this animation in the middle, those things never cross the purple line. Watch this. Never crosses the purple line, still gets out. Everybody see that? So it's just the opposite direction. Is this helpful at all? Except this is Roman two. Passive processes first. I tied it to that first animation of selective permeability. All right, this is what I was talking about here with ATP, so that's down here on the bottom. This is going to be a one of two major topics for lecture exam 1. And you can see the adenine, the ribose, and the 3 phosphate groups. You need to be able to visually recognize the chemical structure of ATP. You also have to know that between the phosphate groups, again, the key here is these bonds are high energy bonds. Not just this one, but this one too. That one still has it. 7 kilocalories per mole, you do need to know that for lecture exam 1. Things that are highlighted, underlined, or bolded. This is an RNA nucleotide. And on Wednesday, I will go over the 4 biologically important macromolecules. I'm going to use the analogy of chains and links. This is a link. This is from your textbook again. If you add a ribose to adenine, it's called adenosine. Don't worry about that. But this is why it's important to take chemistry before physiology. Any questions? Notice that the phosphate group is surrounded by oxygens, and oxygen are all very negative on the outside. And when you get 2 phosphate groups together, it's like trying to get 2 negative ends of a magnet, of 2 different magnets together. You guys ever seen how 2 negative ends, they just repel, right? Just imagine actually getting them together, how much energy it takes to do that. That's what ATPs and ADPs do. Okay, so let's see. I have not referred to the lecture packet much. If you have that lecture packet, we have talked about negative and positive feedback loops in the first couple pages. We did talk about the compartmentalization. That slide that you're going to see several times this semester on page 4. And that has a list of those ions that are different between ICF and ECF, our essay number 9. And then it goes through some transport processes. But here's where we're going to start talking about metabolism. This here, down here in green, is metabolism. One of 2 big topics of lecture exam 1, metabolism. The other one is DNA, and DNA has 2 subtopics. Protein synthesis and DNA replication, cell division. But what we're going to do is we're going to talk about the 4 biologically important macromolecules. The first 3 is listed on page 6. And then on page 7 is a picture of ATP. And what I've actually drawn here, this overall thing down here at the bottom, I know it's kind of small, but you can take a picture of it. This is what is referred to here as the ATP-ADP cycle. Okay, can you kind of see how the arrows are going in a circle? So ATP is on top. If you break off that last phosphate group, look at this. Break off this last phosphate group, there's energy available to do work. So now you have ADP plus P. The reason why you gotta eat nutrients is so you can break them down to put the phosphate, the last phosphate group back on, ADP, so that it becomes ATP. The best, easiest way to understand ATP is think of it like a coin. It's like a coin, and your cells have to build up enough coins to get anything done, to do work. And so when you put that last phosphate group here, the right side of the figures, that's an energy deposit. You're depositing coins. Whenever you want to do any of these guys, you're going to have to do an energy withdrawal and break it off. Do you see that in the figures? Does that make sense of the words on the bottom of the slide?
Mm-hmm.
This is all cellular work. But there's many more. This is just some of them. Everybody got it? Any questions on this? ATP, ADP cycle, that's what's on the whiteboard right there. Do I need to write it down? Because I don't have room for it. But that's what that is, down there in green. ATP, ADP cycle. You already covered active transport. That's one of the cell processes or cell functions that require ATP. But the main topic here, this is a summary statement of what we're going to be talking about with metabolism. There are two forms of metabolism. One is breaking large chains into links. And that would be this. Boom. If you do catabolism of your food, you can put ATP or put the last phosphate group and make ATP. And this is again a big topic for lecture Gem 1. The other one is anabolism, and it's the opposite. It's taking links and building up chains. So one is breaking down, one is building up. Oh, and by the way, right here are the 4 biologically important macromolecules. I'm gonna wait till Wednesday before I write this on the whiteboard because I'm done. Oh. Helpful hint? You guys want a helpful hint for both this class and all your other classes? And the end class? And TEAS test? All of it? Whenever you have paired opposite content. Everybody look at me, man. This is for you, not for me. I aced all my tests. Because I learned this. When I'm studying, I do not want to waste time. When you're talking about a health science career, you gotta take so much fucking classes. Unless you like to go to school for 20 years, you wanna get this thing done. And it's all about points per minute studied. Are you hearing me? Mm-hmm. It's about points per minute studied. You don't wanna waste time. So when you have paired opposite content, only learn one. Only learn one. And just learn the name of the other and know that it's opposite. And that is true for catabolism and anabolism. They are exactly opposite. Learn one, just learn the name of the other and know that it's opposite. And then, in the Scantron, you're not gonna switch them. Because that is not showing points per minute studying. You studied twice as long to learn them both, and you got no points to show for it because you switched them. Yeah, that happens. Mechanical work, sliding filaments, which is actually what is, sliding filaments is where ATP is being burned in bulk transport. Amoeboid movement, cilia, that's all of that kind of mechanical movement requires ATP. Okay, I think I'll just stop there. questions?